So we start with the absolute basics. These are the nutshell type key points in the chapter on enzymes, which, by the way, is your smallest chapter. I think you're down to about 6 pages that you're actually responsible for. If you look at your course outline, it gives you the actual objectives. The full meal deal here, and obviously you are meant to go beyond what I'm presenting here, is best obtained from that textbook and from things like the Raycroft notes, the CD quiz, or the CD presentation. There's a lot of wonderful sources for information. So a little disclaimer here. If you look at your objectives, there are words there like metabolism, enzyme, substrate, coenzyme, activation, energy, etc. These are very important vocabulary words, and you can't really talk about enzymes without having the proper vocabulary. So make sure you're comfortable with the terms listed in that list of objectives. The good news is you're not responsible for any of the chapter material on thermodynamics, endergonic, exergonic, free energy, all that good stuff. Notice there is an objective, though, that comes from way, way beyond the chapter. Page 375, where they're talking about the hormone thyroxine. Thyroxine has an incredible role in terms of metabolism. So yeah, you'll want to check that out as well. So the basics. Enzymes are protein molecules that act as catalysts. At some point in your science career, or if you're taking senior chemistry, you're studying enzymes and catalysts. A biological catalyst is an enzyme. The bottom line is that they are proteins that speed up reactions, but they don't enter into reactions themselves.
Catalysts are substances that speed chemical reactions without being used up in those reactions. In cells, catalysts are usually large globular proteins known as enzymes. There is a portion of an enzyme, its active site, that binds only to a particular molecule, its substrate. The substrate is a participant in the reaction catalyzed by the enzyme. The binding of substrate to enzyme causes a change in the shape of the enzyme, which in turn facilitates the forming or breaking of bonds by the substrate. The product of the reaction is released, and the enzyme is available once again. Because of the fit between substrate and active site, each enzyme is specific for a particular reaction.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
Every reaction that takes place in your body, and there is like 10,000 going on in your cell at any one time, simultaneously require specific enzymes to go from reactants to products. So your diet is incredibly important in terms of making sure you have all of the necessary coenzymes, reactants, etc. Otherwise, you're not going to get the products. If you don't have the products, those products are usually reactants in the next reaction, right? So yeah, I'm just going to show you a chart that I had the joy of being given when I took a biochemistry course. A whole lot of reactions where they show you the reactant, they show you the product, and then in red, you won't be able to see it here probably, but you're seeing the enzymes that catalyze that reaction, that actually make it go. And when we say make it go, yeah, that's an important point. So you are constantly in your body burning sugar, for example. That reaction does not happen outside the body unless you supply a whole lot of energy, a lot of heat, and the heat given off as an exothermic reaction would destroy you as well. So the only way these reactions take place would be with the enzymes that are available. So at some point today, you have probably eaten something, hopefully more than just sugar. But this, of course, is that nasty white substance that, God forbid, you had Fruit Loops for breakfast. That's mainly what is in Fruit Loops. Actually, there was an interesting study done years ago where a group of rats were fed Fruit Loops Another group of rats were fed the box that the Fruit Loops came in, and as you might expect, the rats that ate the cardboard box were actually healthier than the ones that ate the sucrose, Fruit Loops. The point is that when you eat sugar, your body has to do something with that sugar. The only way it can use that sugar is to convert it into glucose. This is an enzyme catalyzed reaction. You will recognize it as hydrolysis. You're adding water, taking a disaccharide, and converting it into the two monomers, glucose and fructose. Point is, that reaction doesn't happen just on its own. That reaction requires a catalyst, and there is the rub. So, take the substrate, sucrose, take the enzyme involved, and again, there's a specific enzyme for each of these reactions, and as you might expect, substrate is sucrose, so the enzyme is sucrase. And sucrase, of course, is a three-dimensional molecule. It's a protein. It has an active site. This is the active site. It's an actual, like a lock and key mechanism where something has to fit into this lock. And at the moment it's empty, but when you have your Fruit Loops, sucrose suddenly is a perfect fit for that active site, and now the enzyme can catalyze the breakdown of that reaction that we showed you just a moment ago. So substrate binds to the enzyme sucrase. That's the first step. Water is available, of course. This is hydrolysis. Substrate is now converted to the products. The products in this case were glucose and fructose, and now your enzyme is again available with its active site, ready for more of those good Fruit Loops. So in terms of enzyme feedback inhibition and etc., you can see what would happen if this enzyme wasn't available. You would never get the products, and of course the body needs the products in order to have any nutritional value from what you're eating. The question is often asked, so what would happen if you didn't have the enzyme available? Can you not break down sucrose without the enzyme? And of course the answer is yes. I have momentarily before you a white powder, a legal white powder. Sugar, and there's no enzyme in that sugar. What I'm going to do is I'm going to break down that sugar. I'll show you. We light the match. We light the sugar. Hopefully you can see this. It's really exciting so far. There we go. Wow. My keyboard will never be the same. Neither will my desk. That is why you don't allow this to take place in your body without an enzyme. It can break down, but the temperature turn you into a piece of charcoal as well. So enzymes are important. They bring down the energy of activation, make that reaction take place at room temperature. It's a biological catalyst. It has to survive in a biological system, a living organism. And don't eat fruits.
For any reaction to occur, even one that releases energy, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, EA. This required energy input represents a barrier that prevents even energy-releasing reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme's substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of a cell's metabolic processes.
Something called reaction specificity or enzyme specificity, it simply means that there's a specific enzyme for each of these very important reactions. Estimates for the number of enzymes in a single cell run from 5,000 to 10,000. Again, see that chart. Enzymes act on substrates. Substrates are what we've always called in chemistry reactants. A plus B gives C plus D. The A plus B are the reactants or the substrates. Just in terms of the terminology here, if the substrate, what's acted on, is a lipid, then the enzyme is called a lipase. And you'll find lipase, an enzyme in your pancreas. Your pancreas. It's part of what's called pancreatic juice. You cannot actually break down lipids unless you have the enzyme lipase. So if you eat that big meal of fish and chips, high fat, you're going to have problems breaking that down unless you have available that enzyme from the pancreas that is going to enter into the duodenum and help in your intestinal breakdown. So the substrate is urea, the enzyme is called urease. If the substrate is called maltose, we call the enzyme maltase. Substrate is called lactose, we call the enzyme lactase. I think you're getting the point. Ase, or something very close to ase. Milk sugar, lactose. This sugar is actually what makes dairy products slightly sweet. Obviously, if you buy sweetened yogurt, you're getting it from sucrose and other God forbidden products like high fructose corn syrup. But yeah, lactose, if you just drink milk, you'll notice it's slightly sweet. A lot of people, I'm not sure what the actual percentage is, but as people get older in our society, there's a lot of people that cannot break down lactose, and they have to take little pills that contain lactase in order to break down dairy products. So for a reaction to occur, the reactants must be brought close together. In other words, a collision must occur. So what is really often called the key model in enzyme substrate reactions is called the lock and key model. A lock fits very close to a key. They fit together in order to make the mechanism work. So it's very similar with an enzyme. The enzyme and substrate fit together like a lock and key at the enzyme's active site. You're going to see a lot of questions that have to do with this active site. Anything that changes that active site, and it's an actual three-dimensional structure. We said that enzymes are proteins, right? So what happens to proteins if you alter things like the pH or high temperature? You're going to change the shape of the enzyme. If you change the shape of the enzyme, the active site is no longer going to fit the key. If you don't fit the key, you're not going to get the product, right? So very important concept. Make sure if you're discussing, say, on the written section of the test, these different factors and their effect on enzyme catalyzed reactions, that you actually use the word active site and say what happens to the active site. Any change to the physical or chemical characteristics of the active site are going to denature the protein. Denature simply means to alter that three-dimensional or quaternary structure of that three-dimensional molecule. You see this actually if you take a dairy product like milk, which contains a protein called casein. If you now add lemon juice or vinegar to that milk, you're going to see denaturation of casein, the protein. You're going to see that sudden separation of the milk proteins into a bunch of floating little bits of cottage cheese floating around in it. Same thing happens when you take albumin, the protein in egg white. When you crack an egg in the morning and you put it in your frying pan, that albumin is actually clear. But as soon as you add heat, notice that your egg white turns this opaque, milky white color. That is because you have denatured the protein. My dear grandmother, God bless her, used to have a saying in the kitchen and I spent a lot of time in the kitchen, I love the kitchen, that a protein boiled is protein spoiled. So one of the key factors in protein denaturation is high heat, not mild heat. If you increase a reaction by, say, up to about 10 degrees, you actually are going to increase that reaction rate significantly because you're increasing the amount of contact between the reactants, or in this case, the substrate, and all of the reactants. So make sure you check out the diagrams in your text, or talk to me and I'll show you some sweet pictures or overheads that we used to use in a bygone era, right? So these are the factors that you have to become comfortable with in terms of their effect on reactions. So temperature, pH, substrate, activators or coenzymes, and inhibitors. There is more here, but I am tempted to go into it in another way. I'll show you these just quickly. This is the effect of temperature on reaction rate. As you increase temperature, notice how the reaction rate increases to a maximum, and then it drops off again to a minimum. We call this maximum the optimum temperature, and every enzyme seems to have optimum temperatures. You have some very strange bacteria that contain enzymes that actually live in hot springs that function best at a very high temperature. At the same time, you've got other enzymes in organisms that live in polar regions that function better at a low temperature. Not surprisingly, optimal temperature within the human body is in fact body temperature, 37 degrees. How about substrate concentration? As you increase the amount of reactants or substrate, notice that the reaction rate is increasing up to a maximum rate where it's going to plateau. And this leveling or flattening out of that reaction rate is because all of the active sites on that enzyme become saturated. They're already plugged with substrate. If you were to add more substrate at this point, you would probably see another increase again in that rate. How about the effect of activators? In some systems, enzymes require or at least function better if molecules such as vitamins are available to function as activators. So eat your vitamins. This is an enzyme alone. This is an enzyme with vitamin K, let's say, or vitamin E. Enzyme and activators. There are reasons in terms of the shape of the molecule itself why often those activators work the way they do. There is something that is basically the opposite of an activator called an inhibitor. What happens if a molecule, should a molecule due to its three-dimensional shape fit into the active site on an enzyme, we would say that it is competitively inhibiting the enzyme in carrying out its normal tasks. The enzyme would not be able to form an activated complex with its specific substrate. So there are substances out there, we call them poisons. And again, I have some poison arrows at home that my dear grandpa, who used to be a missionary in West Africa, brought home from Africa. They are tipped with a paste-like substance. That paste-like substance causes the animal, God forbid a human, to suddenly go into paralysis. And the reason you have that paralysis is because you have an enzyme inhibitor. Suddenly all the enzymes that allow your muscles to function, or some poisons, mercury, etc., heavy metals, they are going to block certain enzymes in pathways for breathing, metabolism, cellular respiration, etc. So poisons work not by magic, but often by being inhibitors to very key chemical reactions of life. All right, just a matter of tying up a few loose ends in terms of things you may or may not see in these enzymes. I mentioned earlier, or showed you earlier in the video, the chart that showed these 10,000 reactions that were catalyzed in a cell simultaneously, and all the intermediaries, the enzymes that made them go. Just in case you are interested, this lovely little chart was again given to us at the end of the unit, and we were asked to identify key molecules. Identify these key molecules in glycolysis and the Krebs cycle. And I mention that only to point out that if you feel that you are being overwhelmed by biochemistry in this course, yeah, you might want to stay away from biochemistry in university. On the other hand, all of these sciences tend to come together, right? So biochemistry is actually an incredibly fascinating area, especially because it's so integral to life. What else can we show you here that's just going to blow your socks off? Yeah, when we talk about enzymes, we're really talking about a process called metabolism. I can write metabolism. Can you write metabolism? Yeah. This is basically the sum of all the chemical reactions taking place in a cell at one particular time or not. And there are two divisions. There are anabolism and catabolism. So if you are familiar at all with bodybuilding and the term anabolic steroids, you kind of get a feel for what anabolism is all about. Anabolism is all those reactions that essentially build up, whereas catabolism is all about those reactions that tend to break down something. Obviously, the reactions that we're talking about are reactions that take place usually as part of cellular respiration and related other biochemical pathways. Respiration, I spelled that wrong, but you know what, you're going to forgive me because you love me, right? Cellular respiration. And of course, you know cells inside out. When you look at this cell, you see the mitochondria. Mitochondria are where cellular respiration occurs. This is just a electron micrograph of liver tissue, and you should recognize these guys as mitochondria. And those are, again, where cellular respiration is taking place. Where you will find a lot of that taking place is in muscles, and you will also see it in the liver. But always, always, always the key molecule in metabolism is going to be ATP. Okay, let's just clean things up a little bit here, okay? Get rid of that. Get rid of that. Get rid of that. Yeah. Do you recognize that molecule? Adenosine triphosphate, ATP. So again, that is the molecule that we're really talking about. A lot of these reactions, if you're in senior chemistry, you will notice that they tend to be of two types: oxidation and reduction. And you have probably learned something about the way to tell the difference, something to the effect that Leo, the lion, says grr. That means is lose electrons, oxidation, or react with oxygen. Gain electrons, we call that reduction. And you can promptly forget all that in this course. It's just that that is essentially what's going on in so many of these reactions. Earlier we talked about the fact that a lot of these reactions can be influenced by what are called inhibitors. This is called competitive inhibition. I could write that, sure. Competitive inhibition. Inhibition. And you, of course, know what inhibit means. Inhibit means to slow down. So we're talking about slowing down the rate of a chemical reaction, if not stopping it altogether. Normally, of course, a substrate is going to bind at an enzyme's active site. This, of course, is Mr. Enzyme. This, of course, is Mr. Substrate. And the two have a lock and key fit. They fit quite nicely together, and they allow the enzyme to function. However, there are molecules out there that function as competitive inhibitors. They fit into the active site instead of the actual substrate. And that's not good, because if you have something else fitting this active site, now the actual substrate can't fit into the enzyme. so the enzyme cannot function, the reaction stops. And if the reaction stops, then whatever product was supposed to happen is not going to be produced, and chances are that product is now going to be the reactant in the next reaction. So that is competitive inhibition. There's also something called non-competitive inhibition, also known as allosteric inhibition, where some inhibitor molecule actually binds at a site other than the active site. And in so doing, notice that it actually changes the shape of the active site. By changing the shape of the active site, the substrate cannot bond to the enzyme. Okay, we're just about there. Take a deep breath. We're going to end this video with an animation, a very important animation on feedback inhibition. All of these reactions that are taking place are multi-step reactions with a product at the end. You cannot have a cell that just keeps producing product, in this case isoleucine. You start with, in this particular case, a substrate, a reactant called threonine, and it goes through a whole bunch of steps, all enzyme catalyzed, and you end up with isoleucine. You can imagine if these 10,000 reactions taking place in a cell were all pumping out their products constantly. Cells would burst instantly. You would die of overdoses of who knows what. So there has to be a way of flipping the switch, a homeostatic mechanism for feedback to say enough. And that mechanism is called feedback inhibition. I'll tell you how it works in this particular case. So we take the substrate threonine. Threonine is not going to become isoleucine all by itself. That's just, not going to happen, right? So what has to happen is you have to have an enzyme present. That first enzyme is enzyme 1, actually has a name called threonine deaminase. Sounds like it's going to remove an amino group or some funky thing like that. But as soon as the substrate binds to the active site of the enzyme, you now get a product. That product now becomes the reactant for the next reaction. And that next reaction requires a second enzyme to become the next product, intermediate B, which requires another enzyme, enzyme 3, to become intermediate C, which requires another enzyme to become intermediate 4, which becomes finally isoleucine. Now the cool thing is, how do you turn off this chain reaction, so to speak? Well, isoleucine now can, via allosteric inhibition, actually fit back into the original enzyme at a site other than the active site, and in so doing, look what it's done to the active site. The active site is now totally messed up, and it's not going to bind with the substrate threonine. You have flipped the off switch. Kind of cool? Yeah. Feedback inhibition. Another chapter where there's a lab that really isn't a lab. Again, for reasons that we don't need to go into, this is a typical enzyme lab, and you will have a paper handout version of this. The idea here is for you to analyze 9 test tubes, all at various pHs and various temperatures, and try to figure out where you're going to get the maximal number of sugar molecules. The reaction taking place here is, as you know, starch, a polysaccharide, being converted to maltose, which is a disaccharide. Both of them, well, this is a sugar. The point is that reaction is catalyzed by a wonderful molecule called amylase. And in this particular case, it's salivary amylase because it's found in your saliva. You might want to try this sometime. Eat a cracker. Start chewing the cracker. You will notice the cracker becomes sweet. And that sweetness is because you now have created a disaccharide. So the question is, if I have a test tube with a bunch of saliva in it, a bunch of starch, and I put it in an ice bath at 5 degrees, I'm going to have like 100% starch to begin with. How much sugar do you think you're going to get at 4 degrees? And I'll tell you, relatively less than you are at other temperatures and other pHs, right? you're going to get some conversion, but the key thing with temperature is you are dealing with basically very few collisions between substrate and enzyme. At that temperature, molecules are moving very slow, so they're not going to start banging into each other and make things happen. On the other hand, take this temperature here, 60 degrees Celsius. What do you think that's going to do to the enzyme salivary amylase? Remember that word denature? At that temperature, you're actually going to destroy the enzyme. You're going to denature it, totally reconfigure its 3D structure so that the enzyme and substrate can't actually meet. So I would say you're going to get 0 sugar there. And again, the same thing happens at these very high pHs. At these high pHs, you're going to end up denaturing again. So the question is, at what temperature and what pH are you going to get much sugar? And I think I've just given you a big hint, but I don't want to totally reveal it. I want you to think about it. What temperature, what pH is going to give you the maximal conversion of starch to maltose? And just in case we missed it, somewhere in all of this, make sure you're familiar with actually being able to draw a lock-in key mechanism. So this would be your substrate, an active site on an enzyme. Notice how they fit together to temporarily form something called an enzyme substrate complex. That is just an incredibly brief period of time, and then it's going to break down, leaving the active site available for more substrate. Again, an enzyme is not changed during the chemical reaction. So
when a metabolic pathway has produced enough end product, it can be temporarily shut down by feedback inhibition. The end product binds to the first enzyme in the pathway. This inhibits the first reaction, and thus all subsequent reactions. The end product is no longer made. In some pathways, the end product binds to the enzyme's active site, blocking binding by the first substrate. In other pathways, the end product binds to an allosteric site, away from the active site. This causes a change in the enzyme's shape, once again blocking the active site. As the end product is used up by the cell, it is no longer available to bind with the enzyme, and the pathway resumes normal operation.